Transcription is the first step of gene expression, in which a particular segment of DNA is copied into RNA by the enzyme RNA polymerase. Both RNA and DNA are nucleic acids, which use base pairs of nucleotides as a complementary language that can be converted back and forth from DNA to RNA by the action of the correct enzymes. During transcription, a DNA sequence is read by an RNA polymerase, which produces a complementary, antiparallel RNA strand called a primary transcript. As opposed to DNA replication, transcription results in an RNA complement that includes the nucleotide uracil in all instances where thymine would have occurred in a DNA complement. Also, unlike DNA replication where DNA is synthesized, transcription does not involve an RNA primer to initiate RNA synthesis. Transcription can be reduced to the following steps, each moving like a wave along the DNA. One or more sigma factors initiate transcription of a gene by enabling binding of RNA polymerase to promote a DNA. RNA polymerase moves a transcription bubble, like the slider of a zipper, which splits the double helix DNA molecule into two strands of unpaired DNA nucleotides, by breaking the hydrogen bonds between complementary DNA nucleotides. RNA polymerase adds matching RNA nucleotides that are paired with complementary DNA nucleotides of one DNA strand. RNA sugar phosphate backbone forms with assistance from RNA polymerase to form an RNA strand. Hydrogen bonds of the untwisted RNA plus DNA helix break, freeing the newly synthesized RNA strand. If the cell has a nucleus, the RNA may be further processed and exits to the cytoplasm through the nuclear pore complex. The stretch of DNA transcribed into an RNA molecule is called a transcription unit and encodes at least one gene. If the gene transcribed encodes a protein, the result of transcription is messenger RNA, which will then be used to create that protein via the process of translation. Alternatively, the transcribed gene may encode for either non-coding RNA genes or ribosomal RNA or transfer RNA, other components of the protein assembly process, or other ribozymes. A DNA transcription unit encoding for a protein contains not only the sequence that will eventually be directly translated into the protein but also regulatory sequences that direct and regulate the synthesis of that protein. The regulatory sequence before the coding sequence is called the 5' untranslated region, and the sequence following the coding sequence is called the 3' untranslated region. Transcription has some proofreading mechanisms, but they are fewer and less effective than the controls for copying DNA. Therefore, transcription has a lower copying fidelity than DNA replication. As in DNA replication, DNA is read from 3' end to 5' end during transcription. Meanwhile, the complementary RNA is created from the 5' end to 3' end direction. This means its 5' end is created first in base pairing. Although DNA is arranged as two antiparallel strands in a double helix, only one of the two DNA strands, called the template strand, is used for transcription. This is because RNA is only single-stranded, as opposed to double-stranded DNA. The other DNA strand is called a coding strand, because its sequence is the same as the newly created RNA transcript. The use of only the 3' end to 5' end strand eliminates the need for the Okazaki fragments seen in DNA replication. In virology, the term may also be used when referring to mRNA synthesis from a RNA molecule. For instance, the genome of a negative-sense single-stranded RNA virus may serve as a template to transcribe a positive-sense single-stranded RNA molecule, since the positive-sense strand contains the information needed to translate the viral proteins for viral replication afterwards. This process is catalyzed by a viral RNA replicase. Major steps: transcription is divided into pre-initiation, initiation, promoter clearance, elongation and termination. Pre-initiation, in eukaryotes, RNA polymerase, and therefore the initiation of transcription, requires the presence of a core promoter sequence in the DNA. Promoters are regions of DNA that promote transcription and, in eukaryotes, are found at -30, -75, and -90 base pairs upstream from the transcription start site. call promoters are sequences within the promoter that are essential for transcription initiation. RNA polymerase is able to bind to call promoters in the presence of various specific transcription factors. The most characterized type of call promoter in eukaryotes is a short DNA sequence known as a Tata box, found 25 to 30 base pairs upstream from the TSS. The Tata box, as a call promoter, is the binding site for a transcription factor known as Tata binding protein, which is itself a subunit of another transcription factor, called transcription factor 2D. After TFID binds to the Tata box via the TBP, 5 more transcription factors and RNA polymerase combine around the Tata box in a series of stages to form a pre-initiation complex. One transcription factor, transcription factor 2H, has 2 components with helicase activity and so is involved in the separating of opposing strands of double-stranded DNA to form the initial transcription bubble. However, only a low, or basal, rate of transcription is driven by the pre-initiation complex alone. Other proteins known as activators and repressors, along with any associated coactivators or co-repressors, are responsible for modulating transcription rate. Thus, pre-initiation complex contains, call promoter sequence, transcription factors, RNA polymerase, activators and repressors. The transcription pre-initiation in Archaea is, in essence, homologous to that of eukaryotes, but is much less complex. The archaeal pre-initiation complex assembles at a Tata box binding site. However, in Archaea, this complex is composed of only RNA polymerase II, TBP, and TFB. Initiation. In bacteria, transcription begins with the binding of RNA polymerase to the promoter in DNA. RNA polymerase is a core enzyme consisting of 5 subunits, 2I plus or minus subunits, 1I squared subunit, 1I squared subunit, and 1I per mil subunit. At the start of initiation, the core enzyme is associated with a sigma factor that aids in finding the appropriate -35 and -10 base pairs downstream of promoter sequences. When the sigma factor and RNA polymerase combine, they form a holoenzyme. Transcription initiation is more complex in eukaryotes. Eukaryotic RNA polymerase does not directly recognize the call promoter sequences. Instead, a collection of proteins called transcription factors mediate the binding of RNA polymerase and the initiation of transcription. Only after certain transcription factors are attached to the promoter does the RNA polymerase bind to it. The completed assembly of transcription factors and RNA polymerase bind to the promoter, forming a transcription initiation complex. Transcription in the Archaea domain is similar to transcription in eukaryotes. Promoter clearance. After the first bond is synthesized, the RNA polymerase must clear the promoter. During this time there is a tendency to release the RNA transcript and produce truncated transcripts. This is called abortive initiation and is common for both eukaryotes and prokaryotes. In prokaryotes, abortive initiation continues to occur until an RNA product of a threshold length of approximately 10 nucleotides is synthesized, at which point promoter escape occurs and a transcription elongation complex is formed. The I-fluorine factor is released according to a stochastic model. Mechanistically, promoter escape occurs through a scrunching mechanism, where the energy built up by DNA scrunching provides the energy needed to break interactions between RNA polymerase holoenzyme and the promoter. In eukaryotes, after several rounds of 10-NT abortive initiation, promoter clearance coincides with the TFIIH's phosphorylation of serine 5 on the carboxy terminal domain of RNAP2, leading to the recruitment of capping enzyme. The exact mechanism of how CE induces promoter clearance in eukaryotes is not yet known. Elongation. One strand of the DNA, the template strand, is used as a template for RNA synthesis. As transcription proceeds, RNA polymerase traverses the template strand and uses base pairing complementarity with the DNA template to create an RNA copy. Although RNA polymerase traverses the template strand from 3' to 5, the coding strand and newly formed RNA can also be used as reference points, so transcription can be described as occurring 5' to 3. This produces an RNA molecule from 5' 3, an exact copy of the coding strand sugar where DNA has deoxyribose in its sugar phosphate backbone. mRNA transcription can involve multiple RNA polymerases on a single DNA template and multiple rounds of transcription, so many mRNA molecules can be rapidly produced from a single copy of a gene. Elongation also involves a proofreading mechanism that can replace incorrectly incorporated bases. In eukaryotes, this may correspond with short pauses during transcription that allow appropriate RNA editing factors to bind. These pauses may be intrinsic to the RNA polymerase or due to chromatin structure. Termination. Bacteria use two different strategies for transcription termination: row-independent termination and row-dependent termination. In row-independent transcription termination, also called intrinsic termination, RNA transcription stops when the newly synthesized RNA molecule forms a GC-rich hairpin loop followed by a run of Us. When the hairpin forms, the mechanical stress breaks the weaker U-DAR bonds, now filling the DNA-RNA hybrid. This pulls the poly-U transcript out of the active site of the RNA polymerase, in effect, terminating transcription. In the row-dependent type of termination, a protein factor called Rho destabilizes the interaction between the template and the mRNA, thus releasing the newly synthesized mRNA from the elongation complex. Transcription termination in eukaryotes is less understood but involves cleavage of the new transcript followed by template-independent addition of As at its new 3' end, in a process called polyadenylation. Inhibitors. Transcription inhibitors can be used as antibiotics against, for example, pathogenic bacteria and fungi. An example of such an antibacterial is rifampicin, which inhibits prokaryotic DNA transcription into mRNA by inhibiting DNA-dependent RNA polymerase by binding its beta subunit. 8-hydroxyinulin is an antifungal transcription inhibitor. The effects of histone methylation may also work to inhibit the action of transcription. Transcription factors. Active transcription units are clustered in the nucleus, in discrete sites called transcription factors or euchromatin. Such sites can be visualized by allowing engaged polymerases to extend their transcripts in tagged precursors and immunolabeling the tagged nascent RNA. Transcription factors can also be localized using fluorescence in situ hybridization or marked by antibodies directed against polymerases. There are 10,000 factors in the nucleoplasm of a HeLa cell, among which are 8,000 polymerase II factories and 2,000 polymerase III factories. Each polymerase II factory contains 8 polymerases. As most active transcription units are associated with only one polymerase, each factory usually contains 8 different transcription units. These units might be associated through promoters and/or enhancers, with loops forming a cloud unregistered trademark around the factor. History. A molecule that allows the genetic material to be realized as a protein was first hypothesized by Section Jacob and Jacques Monod. Severo Ochoa won a Nobel Prize in Physiology or Medicine in 1959 for developing a process for synthesizing RNA in vitro with polynucleotide phosphorylase, which was useful for cracking the genetic code. RNA synthesis by RNA polymerase was established in vitro by several laboratories by 1965. However, the RNA synthesized by these enzymes had properties that suggested the existence of an additional factor needed to terminate transcription correctly. In 1972, Walter Fiers became the first person to actually prove the existence of the terminating enzyme. Roger D. Kornberg won the 2006 Nobel Prize in Chemistry for his studies of the molecular basis of eukaryotic transcription. Measuring and detecting transcription. Transcription can be measured and detected in a variety of ways. Nuclear run-on assay measures the relative abundance of newly formed transcripts. RNase protection assay and chip of RNAP detect active transcription sites. RT-PCR measures the absolute abundance of total or nuclear RNA levels, which may however differ from transcription rates. DNA microarrays measures the relative abundance of the global total or nuclear RNA levels. However, these may differ from transcription rates. In situ hybridization detects the presence of a transcript, MS2 tagging, by incorporating RNA stem loops, such as MS2, into a gene. These become incorporated into newly synthesized RNA. The stem loops can then be detected using a fusion of GFP in the MS2 coat protein, which is a high-affinity, sequence-specific interaction with the MS2 stem loops. The recruitment of GFP to the site of transcription is visualized as a single fluorescent spot. This new approach has revealed that transcription occurs in discontinuous bursts, or pulses. With the notable exception of in situ techniques, most other methods provide cell population averages, and are not capable of detecting this fundamental property of genes. Northern blot, the traditional method, and until the advent of RNA-seq, the most quantitative, RNA-seq, applies next-generation sequencing techniques to sequence whole transcriptomes, which allows the measurement of relative abundance of RNA, as well as the detection of additional variations such as fusion genes, post-transcriptional edits and novel splice sites, reverse transcription. Some viruses have the ability to transcribe RNA into DNA. HIV has an RNA genome that is reverse transcribed into DNA. The resulting DNA can be merged with a DNA genome of the host cell. The main enzyme responsible for synthesis of DNA from an RNA template is called reverse transcriptase. In the case of HIV, reverse transcriptase is responsible for synthesizing a complementary DNA strand to the viral RNA genome. The enzyme ribonuclease H then digests the RNA strand, and reverse transcriptase synthesizes a complementary strand of DNA to form a double helix DNA structure. The cDNA is integrated into the host cell's genome by the enzyme integrase, which causes the host cell to generate viral proteins that reassemble into new viral particles. In HIV, subsequent to this, the host cell undergoes programmed cell death, or apoptosis of T cells. However, in other retroviruses, the host cell remains intact as the virus buds out of the cell. Some eukaryotic cells contain an enzyme with reverse transcription activity called polymerase. Polymerase is a reverse transcriptase that lengthens the ends of linear chromosomes. Polymerase carries an RNA template from which it synthesizes a repeating sequence of DNA, or junk DNA. This repeated sequence of DNA is called a telomere and can be thought of as a cap for a chromosome. It is important because every time a linear chromosome is duplicated, it is shortened. With this junk DNA or cap at the ends of chromosomes, the shortening eliminates some of the non-essential, repeated sequence rather than the protein encoding DNA sequence, that is farther away from the chromosome end. Polymerase is often activated in cancer cells to enable cancer cells to duplicate their genomes indefinitely without losing important protein-coding DNA sequence. Activation of polymerase could be part of the process that allows cancer cells to become immortal. The immortalizing factor of cancer via telomere lengthening due to polymerase has been proven to occur in 90% of all carcinogenic tumors in vivo, with the remaining 10% using an alternative telomere maintenance route called ALT, or alternative lengthening of telomeres. See also, Crick's central dogma: DNA is transcribed to RNA, which is translated to polypeptides, eukaryotic transcription, gene regulation, prokaryotic transcription, RNA polymerase, reverse transcription, process viruses used to make DNA from RNA, splicing, process of removing introns from precursor messenger RNA to make messenger RNA, translation, process of decoding RNA to form polypeptides. References. External links. Interactive Java simulation of transcription initiation. From Center for Models of Life at the Niels Bohr Institute. Interactive Java simulation of transcription interference, a game of promoter dominance in bacterial virus. From Center for Models of Life at the Niels Bohr Institute. Biology animations about this topic under Chapter 15 and Chapter 18, Virtual Cell Animation Collection, Introducing Transcription. Easy-to-use DNA transcription site.